always like to begin our class every week with a scripture. There are so many of them in the Bible that reveal the truth that is so important. I think it's the most fundamental truth, and that is God is our creator. He's much more, but he is that in such a very important way. Revelation 4, verse 11, you are worthy, our Lord and our God, to receive glory and honor and power, for you created all things. They came into existence and were created because of your will. So one of the reasons we come together and praise God and magnify him is because he's our creator. He is worthy of glory. He made everything. And he didn't just make everything sort of vanilla. He made everything wonderful. And he made everything lovely. And he made everything because he loves us. And so these are truths that are fundamental to our faith. And if he hadn't have created all the wonderful things we see out here as also an evidence of his existence, we would have plenty to be able to understand that he is our God. And especially as we look at the scriptures and see the evidences for their inspiration, it's a complete set of evidence that we're not looking at this particular time, but that I think are the strongest evidences for God and for his inspired word and for his son, Jesus Christ. So praise God for his goodness and for his creation. He is worthy of our praise. I wanted to mention that our PowerPoint slides are available, as I've mentioned a few times, and I actually started making a contents for them. It's up to 3 pages of just contents, and it's a clickable content. So if you want to go to any of the material we've covered, you can just click on one of these things and go right to that if you're looking for something. Briefly, we've covered some introductory material. Part 1 was evolution, the evidence, which really there is none that holds up. And so you might remember some of these subjects we've been through in the past here and covered a number of weeks ago. And then page 2 gets into part 2, creation evidences, which were, of course, the main part of our study. Looked at the human body and the eye and the ear and some of the other mechanisms that are wonderfully amazing in our bodies. And then looked at a number of creatures of design. That we spent some time seeing that the only way they could come into existence is if they were designed by our God. Evolution just falls miserably short of any explanation of how any of those creatures could have come into existence. And then now we are in a study in design in molecular biology, something we couldn't have done 100 years ago because they didn't have some of the sophisticated machinery that we have now, the devices, the microscopes that can magnify, as much as they do. But it's still, as we look at things around us in normal resolution, it's wonderful. It's amazing. When you look at things in a microscopic level, it's just as amazing. The creatures, the little robots, as Andrew called them, that he made and designed so that all this thing that we see on this level, our bodies and all the plants and the animals, can function the way they do. So we're going to just quick review. Remember, Darwin, when he came up with this theory, he didn't have any idea. In fact, if they had some of the tools that we have today, he likely wouldn't have gone through and published his Origin of Species. He would have seen, just like a lot of other scientists today have seen, that these things are too complicated. They're too amazing. They're too sophisticated to be able to come about by a system of random chance, which is what mutation uses. So just random chance and selection. So on the left there, that little blob of jelly is all they could really see with the microscopes they had of their day. But today, we can magnify not only 800 times, we can magnify 50 million times. So even at this actually low resolution of the cell on the left, a couple thousand magnification, we understand there's so much sophistication just in each cell. And we have about 50 trillion cells, and they're all amazingly complex and sophisticated and perfectly coordinated to do the job of our bodies. Yes, and I'm going to remember your name. It's Spencer. Yes, Spencer, yes.
A pediatrician friend of mine told me that every mutation is actually detrimental to the body.
Yep.
And evolution says that mutation has created all this complexity. How did it maintain
You're right.
without falling apart during all these millions of mutations. It's irresponsible and it's impossible to have evolution. It has to be creation.
Yes, exactly. I appreciate that. It's exactly true. I believe in devolution. I really do. Things are devolving. In fact, that's consistent with the first couple chapters of Genesis. We're under a curse. Things are corrupting. That's why mutations happen, because a copying error that would never have happened if we still were in the Garden of Eden, but we aren't. We're in a corrupted world, a world that needs to be redeemed spiritually, and one day we'll be redeemed physically when we all come into glory with him in heaven. Thanks, Spencer. Appreciate the input there. So here's a graphic we've used of the cell, the complicated mechanisms. And just looking briefly here at what we've covered so far, we looked at these rotary generators in the cell that are responsible for making all the power, the energy that all the tissues and other little machines need to run. And these things are extremely sophisticated, microscopic. Again, they're all bumpy looking like that, not because God doesn't know how to make things smooth, but because that's as small as matter gets. So those are protons, the little bumps you're seeing, and parts of, or at least atoms you're seeing here that are making up these proteins that make up this little generator. And so we looked at something, okay, just a second here. On, like these little bacteria motors that spin, and there on the top right is an actual electronic microscope picture of this motor that's a propeller that drives bacteria around. Is bacteria good or bad? You're supposed to say yes. Yes. Okay. We got helpful bacteria, and we've got harmful bacteria. So it drives these things around, and that's not overselling it. That little graphic that's spinning there. They are little motors that run around about 20,000 RPM. They spin and drive these things around. Amazing machines. See if I can get this next one here. The little cargo trucks of the cell. These little guys, kinesins and dyneins, are little machines, robots, Andrew called these, that run around doing different jobs, carrying proteins to where they're needed, moving things from one side of the cell to the other, taking things outside the cell. And they're controlled and made by the sophisticated process of the cell. So these things are just amazingly wonderful things. And the thing is, we're not here to teach everybody about microbiology. We're not. But to realize that when you look at these kinds of things, these are machines like any normal resolution machine that man made. Far more sophisticated, actually, than what we've made, and they prove something. They're evidence for something. They couldn't fall together by accident. They were made. They were designed. They were made by God. And all life forms have these little machines. And we're just scratching the surface. There's 20,000 molecular machines, protein molecular machines that we have in nature. And we're just looking at a few of them here. So that's kind of as far as we got last time. We looked at a few more than this, but these are the ones that we've looked so far. Yeah, Mike. Wait a sec. Oh, you got a mic. I'm sorry.
Yeah.
Mike has a mic.
The idea that things by mutation are improving is akin to the idea that if you got a flat tire on your car, all you should do is wait until the other three go flat, and then it grows wings. It makes no sense. It doesn't fit in with the pattern of things in general. The second law of thermodynamics, which says everything is running down. And the only reason that the things have the quality and the excellence that they do is that God initially input those things into the creation, and
Amen.
that's why He said it was very good, and it was indeed. We're seeing how very good it all was.
Amen. Wonderfully good and amazing, and only way that it could be here is because of because of our God. Thanks, Mike. Terry. Hey, Matt, Terry over here. Rob. Okay, we'll get over to her. We want to have it on the mic just so people who are listening in and who are maybe listening to this later on can have the input. Terry.
I find it interesting at what time in history God has allowed us to see this magnification.
Yeah.
In David's day, they didn't have motors like this. They might have had a water mill, but they would not have comprehended this because God's given us the knowledge and technology in our machinery that they wouldn't have had back then.
That's a good point. I never thought of that. It wasn't until we all already developed our own man-made machinery in the last, I don't know, 100, 200 years, that we could maybe even appreciate this kind of machinery. Back in David's time, it was plenty for him to see what was around him. Certainly because he wrote some of these Psalms we've been reading about God being our creator and how obvious it is. But good point. Yeah, we have a better ability to be able to understand this type of information, which God has allowed through the technology of microscopes today. And it's so funny, the stronger evolution is taking hold of our world, our civilization, the more evidence God is allowing us to see of how amazing his creation is. So yeah, thank you for that. Okay, I want to look a little closer at this last machine. This is that ribosome that makes proteins. It's the only thing in nature that makes proteins, is this little machine. Remember now, that little chain going through the bottom is what's called a messenger RNA, and basically it copies a section of DNA that has the information to make whatever proteins the cell wants to make. And it comes over and it runs through the bottom of that thing and telling it every time, okay, bring this amino acid, bring this amino acid. And then it builds out the back the protein that it wants. Now you got to put your thinking caps on here for a minute, because I'm going to ask you a question that's pretty important. So this thing runs and these amino acids are brought in by transfer RNA. All this protein is being assembled out the back. Now it looks all like a little chain. It gets folded up into a machine or mechanism of some sort and goes off and does its job somewhere. So here's the question for you. This ribosome is made of about 30 proteins itself. So my question for you is, how is a ribosome made? How is a ribosome machine? How is this machine made? It itself is made of 30 proteins.
You have more than one in your cell. It's fine. Don't make each other up.
They do make it. Okay, let's go back to the beginning. Here's the question. Not only is this thing amazingly designed and sophisticated, it's obvious, but the question comes up now for evolution. How did the original ribosome machines come about? They themselves are made of proteins. So you're right, and your answer is that other ribosomes make the proteins that go in the new ribosome. That's how it works today. But what about the beginning? You've got a protein machine that makes, the only thing that makes proteins, but it needs proteins to exist itself. So it's basically what's called an impossible paradox. You can't come up with something. It had to be in place. The only explanation is a designer who designed it and made it complete, because these things are so interrelated. It couldn't build itself, the first ribosome. You know, it didn't have the raw materials. It didn't have, ribosomes need, are built of proteins. So I'm not sure if I'm explaining that quite well. A ribosome is built of proteins, but proteins are made by ribosomes. It's an impossible circle. Of course, today it's not, because we have all this everywhere in nature, but originally, in the beginning, how did it all start? Evolution has no explanation, and believe me, go and look and see. They don't know. Those who champion that particular theory don't know how this could have happened. Okay, one more question. Here's a machine we didn't spend any time looking at. It's called RNA polymerase. I'm making a point about this one too. It's the same kind of a point. There's an impossible paradox that comes up. This little machine runs along the DNA molecule, the information molecule, and spits out at the top this little RNA molecule that has the information that it sends on to this little coded piece of information here. And these are codes, just like a computer code. There's different order of these codes. That thing spitting out the top is actually what this thing is here coming through the ribosome. So ribosome can't make anything without this little information RNA coming through. But this is the only guy that actually produces that RNA strand that comes over and gets copied, or actually that not gets copied, it gets, the ribosome turns it into proteins. So, do you see the problem? What is this little machine made of that produces the RNA? Give me a guess. Proteins, a number of sophisticated proteins. But wait a minute, the ribosome couldn't produce the proteins to make this machine without the RNA molecule, which is only made by this machine. So you've got this impossible paradox again. We're talking about origins here. Well, let me say, we're actually talking about design. But now looking back, how did this thing come about to begin with? You couldn't have a ribosome make anything unless there was an RNA polymerase machine producing that RNA strand, but you couldn't have that unless you got the proteins that the ribosome makes. Impossible paradox. Evolution has no answer for this. The things that I've read, there is no good theory that explains this. And everything in life, plant, animal, man, has these same kind of machines. It's what runs everything. So, if you've got an answer to that dilemma that would help evolution, let me know afterward. Not that I'm for evolution, but I want to know what best answer they can give. I haven't found any. Okay, moving on to really our last one that we're going to look at in any detail. It's one of the most complicated molecular machines that we have in our bodies. It's called the spliceosome. Here's what it does. It removes introns and inserts exons into precursor messenger RNA. Test next week, so make sure you memorize that. What does that mean? Better explanation, it's like one of these in our world. It basically takes not film, but RNA, that strand that we're talked about, and cuts out certain pieces and splices that rest together. Just like a tape machine. Like when people go out and make movies, they go out and take just, miles and miles of tape of movies, and then they come together in a splicing room and cut out the parts they don't want and then keep the parts they do. So that's pretty easy to understand. Well, that's what this molecular machine does. And it has to, because the DNA, let me go on to the next slide here. We've got our little machines going. There's our ribosome that makes the proteins. There's our little polymerase that makes the RNA. The RNA that it copies from DNA has not only parts that code for proteins, but it has other informational sections, all in line. Boom, 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 boom. And so as this RNA can't go directly to the ribosome, it has to be spliced first. I left out that step before I explained because it gets too complicated. But that's what this machine, before it goes to the ribosome to make all these proteins, it has to run through the spliceosome. Again, we're not trying to teach molecular biology, just to understand the complexity of what God has made on this level. And so, this little piece of RNA can't just go from this particular machine. Let me get my thing turned around here. Directly to here. It's got to go through this guy first, and then it comes over here. Look, a little bit of an animation that shows this. The first part of it is going to be the thing that actually copies the RNA. You'll see that, and then and this is not the best video, but let's see.
Before the gene can be translated into a protein, the intron non-coding sequences must be removed.
So here's the spliceosome, that thing that comes out. You can't just send it over to be making proteins. You have to go through the splicing mechanism that pulls out the parts that don't code for proteins. They have other uses, those particular strands, but not for building proteins. Okay.
The process of exon removal is catalyzed by a large molecular complex called the spliceosome. The spliceosome is highly dynamic, involving sequential binding and release of as many as 300 distinct molecules, of which only a few are represented here. These remarkable dynamics are largely the same in all eukaryotes, from yeast to humans.
These are present in all cells, he's saying here, man and animal cells, and eukaryotes, a certain kind of a cell, the most common. Okay, so that's just to show you, again, the interconnection between these machines that goes on in our body, trillions of these things going on at one time in our cells, And they're very sophisticated and necessary for for our bodies to continue and to copy cells. I'm going to play part of another video that shows this, and I'm going to go through this video fast if I can. You can always go online and look at this in detail, but it shows you how complex this particular machine is. Hey, Dan, if you had brought a car over, sometime and say, hey, Bill, let's go for a ride. I got a special car. Really, say if you came over someday to show me a car that you said was really special. Dan's a mechanic, for those that don't know. And he said, yeah, come for a ride. This car is really cool. And we go out and we drive along. It looks like any car to me. But he said, look what this can do. And he would go along and the wheels fly off. And here out from nowhere, zoom, come in some more parts. And we climb a tree. And I'm going, I've never seen a car like this. This is amazing. And then we go along further. Oh, did I do something? Oh, okay. I heard the knock, knock, knock. Okay. Come on in. And we come to a body of water. He says, and I go, look, we can't go across there. We got wheels on this car. At least we switch back to wheels. But he says, don't worry. Boom, the tires pop off and comes in this hole. And now we go across the lake. And I'm thinking, boy, Dan, you're an amazing mechanic. I never knew you knew how to do this. And all this is happening while we're just traveling along. Parts pop off, more parts come in, parts pop off, more parts come in, until he gets me to where we're going. You got a car like that? No. Okay, I didn't think you did. Okay, well, this machine is like that. And I'm going to show you a little bit, and if I can get to this.
Initial events require.
Don't worry about listening to this guy.
5' splice site and branch point
turn up the volume a bit,
by
maybe.
U1 and U2 SNRs, notably involving base pairing.
It's showing, and let me stop it for a minute. That little red thing here going along, again, is the RNA molecule, but it's going to take out a section that's not needed to make a protein. And it does this through this machine does this through a multi-modular machine that comes and goes as these parts are needed. Yeah, Dan, you do have a car like that.
Well, it's very interesting, how well we're created, how God created us. And you look at everything around us, and just in the automotive industry, and you go back to like 1930 or even the 20s, where you actually had to retard the timing, pull this lever for the choke, twist the air fuel mixture just to start your car up. And it just seems like the more we figure out how our body works, now we have sensors and actuators and solenoids that do all this for us. Just like this here, we developed vehicles that will go across water or drive on land. Mm-hmm. I even think we have a car that'll drive down the road and can fly a little bit too.
Yeah.
So we develop, we are making this machinery just like us. Just like, you look at an excavator, how it digs. When we didn't have anything, we dug with our hands. Yeah. You take a shovel and if you want to dig a trench, you're pulling it towards you. Mm-hmm. So everything how we're designed, that's how we design everything around us. We have sensors in our cars that you drive down the road and it tells you that someone's over here, just like you can sense someone coming up behind mm-hmm. you. Mm-hmm. So it's just like all these, we got air pressure sensors in our tires. So, if you stub your toe, you stubbed your toe. So if your tire's flat, the little light comes on and says, hey.
So what did it take to make all that? That just fell together by chance, didn't it?
Well, when we figure out how our body works.
Right.
It's just like I look at like when the computers come out, mm-hmm. we finally, and probably in the 40s, I'm guessing, when we were chopping up brains, and especially in Germany, okay. we're doing all these experiments. You figure out how the body worked, and you can kind of replicate that a little bit into our everyday lives.
A lot of that's been done.
Yeah,
from the things that are in the body and the machinery that we have, a lot of things are copied by engineers today and making different devices.
Right. It's basically us humans in a different form.
Yeah, which of course is God who designed us and made us.
It's
and then
perfect.
we take his idea, we steal it, we plagiarize his ideas,
Right? Exactly.
is what we do.
We're just copying God's
Yeah.
what he created.
Exactly. Appreciate that. So Dan doesn't have a car like that, like this particular machine. I'm going to see if I can, because this thing's like a 5-minute video and I don't want to play all of it because it's boring. But, oh, not that boring. Let me go back. Let me start it.
Initial
And let me see if I can go through it. And the thing to get is all the parts leaving and the parts coming and the parts going as it does its job of splicing this RNA. Let me see if I can.
RNA, which are stabilized by protein
Not going to
components.
work.
In the
If I do it enough, it might
resulting
work.
A complex or pre-spliceosome, the U1 and U2 SNRs are linked, but they may be mobile
keep
relative
trying
to
here.
each other.
Give me a minute.
Next, the tri-SNRP particle will associate with the A complex, followed by RNA helicase PRP28 and Res complex, which begins to function to stabilize the pre-mRNA.
There's a way that works sometimes to fast forward these videos. Sometimes it doesn't work.
Forming a
I
suitably
really don't want to sit
inclusive
here
cluster,
and watch
the
the whole
pre-D
thing.
complexes, relaxed interfaces
I
between
can't
proteins
seem
and
to get that to
RNAs,
do
they
that.
facilitate remodeling
Oh, let me
of
see here. Hold on,
pre-mRNA
just-
splice
just a
site
second.
reactants and concomitant construction of the active.
Oh, wait a minute.
Initial events require recognition of 5'
No, not
splice
gonna get
site
to work.
and branch point sequence.
Well, if you really want to see how that works, you can go later and just get the PowerPoint slides that we're making a variable and watch that. What I just have to explain to you. It's an amazing thing. This particular machine doesn't just sit there with the many components it has when it starts its work. It switches them out, other ones come in, and then when it's all done, it has machines come in that disassemble it and recycle the parts. I mean, Dan can't even make a car like that, and yet God does. And it's just tremendously amazing what he does. Kurt.
I just wanted to expand on what Dan was saying about how we look to God's design to design everything around us. You know, things that we build, you know, we get our design ideas from what God has created. And
You don't have a book like that, do you?
Oh yeah, I think it's called Made in Heaven by
That's
Ray
right,
Comfort.
you do. He gave it to me actually too.
Yeah. It's really fascinating because it just shows you everything that we design really comes from God's design. I mean, because God has ultimate wisdom. He knows how things works efficiently, and we're learning from that design. So yeah, I just wanted to mention that book.
Yeah. And I, by the way, I looked through the whole thing. It's really good. Not sure if we'll have time to include the stuff, but what it says is what Dan was saying too, is that it looks at a number of the advancements in science and technology that we copied from God's designs and borrowed and modified some way. And so we're not as smart as we thought we were. We didn't come up with all those ideas. We looked and saw what God had made, and we said, yeah, we can make something like that. So still can't have it make its own self and repair its own self the way that God's machines do, but still, that's great. Yeah, it's a good resource there that Kurt has. Mike, did you have your hand up?
Yeah, so it would be amazing enough if we just had one of these machines, molecular machines in our bodies, you know, functioning as it does. But when we consider that we have these highly complex molecular machines interconnected one with another in order to accomplish a definite purpose, you know, we literally have a factory in every cell that is, you know, set up to produce something. And, you know, we know that in our world, if we wish to produce anything, it requires the coordinated efforts of many parts. And so why is it when we look at our own bodies and we look at the cells that we cannot see, I'm speaking, of course, in general, cannot see that there must be someone who designed them to be that way. who brings them all together?
Chiefly, and you know the answer to that question, is because we don't want to. And I'm speaking generically too. We don't want to be accountable to God. It's the only rational explanation, but if I can come up with something else that, might answer that, even though it's ridiculous and foolish, as God says, I'd prefer that answer, is what happens a lot of time, and you know that. So yeah, good point. Okay, so here we got this spliceosome. This multi-modular machine continually switches out its various component parts as it carries out its function. An amazing example of molecular engineering. If you don't know what the mousetrap means, go back and listen to the other classes that we did. It's just a simple example of irreducible complexity. None of the parts by themselves, if they evolved over millions of years, would have done anything, and they wouldn't have survived to produce the whole mousetrap if its other various parts didn't have some significance before all of it could have evolved. These things are so much more complicated, and the reality is the same thing is true. They're too complex to come about by a slow process of mutation and natural selection. Yes?
That's the crux of the matter from what you're showing us, and I'm not gonna understand molecular biology, but the takeaway is this, the irreducible complexity. None of these parts can exist without the other parts.
Yeah.
They can't even come into being.
No.
And evolution says mutation brings about these different parts. Well, you could try to explain it with one part, but where do you get all the other parts that function together? And they can't exist without the other.
Exactly.
So you got multiple parts that can't exist without the other. And it becomes, the foolishness becomes what they say. The wisdom is creation. God made it.
Amen. It is.
In its complexity.
And that's part of the thing we need to understand. Not only are these things individually too complicated to come together, but there's no explanation for evolution how that any number of them could have somehow evolved at the same time to make the first living cell. And we'll look at this argument in a bit when it comes to DNA, but the thing is that that's completely impossible. No evolution even tries to explain that, at least not in any way that I can see that makes any sense. They just say, we know it had to happen. We know it just had to happen some way. We'll figure it out one of these days. And hopefully they will figure out the answer. But it's not evolution, it's God. Thanks, Spencer. Okay, moving on. If we had time, we'd look at this guy here. Amino Aacl tRNA synthase. It's another machine. You remember that guy that brings those amino acids into the ribosome so it can build the proteins? Well, this is the machine that puts that little amino acid on that tRNA so it can go over and put it in the ribosome. It's a sophisticated molecular machine. In fact, the next thing says, attaches the appropriate amino acid to an RNA strand. Again, you need all these things to happen. Those things just don't float by the ribosome when they want to. These guys make the part of the mechanisms that bring them. Okay, we could look at the bacteria odsophen, if that's how you pronounce it, which it probably isn't. Compact molecular machine that uses the energy of sunlight. Oh, a whole different dimension there as far as sophistication. The energy of sunlight to pump protons across a membrane. Why? Go read a book about this if you want to learn, but it's a sophisticated machine that does this. We could look at myosin. Myosin, a very sophisticated particular molecular machine, a motor that moves along a track to form the basis of muscle movement. Why am I so strong? Here you go. Here are these guns up here. It's because of this guy. Or I can also have weak muscles, which is actually I do, right? I don't have very strong. And they're amazing how they work. It doesn't just happen like a rubber band. They move along a track and they contract the muscle, and it's a mechanism that works this way. It also, this particular thing has double job. It also can transport cargoes within the cell, like a dynein or a kinesin that we looked at before. Proteasome. This is a molecular machine. Let's see what it says here. A molecular machine whose function is to degrade and destroy proteins that have been misfolded. So we haven't talked about the air checking capability of cells. They have all sorts of air checking. Make Microsoft look ridiculous when it comes to, it looks like he doesn't have anything. Air check, air check. But when it finds out that a protein's been misfolded, this guy comes along and that protein goes in and comes out with the basic amino acids and gets recycled or thrown away. Sophistication upon sophistication upon sophistication is what you see when you look at the molecular level. It's amazing. And again, I'm going to cycle through these. We saw these already, but we're going to read somebody and what he has to say real quickly as we look at these things. Just a second. So here's the same thing. All these machines, we're wrapping this part of it up. So many more. If you're interested, go look them up. And just see molecular machines in the body, and you'll get this kind of information. So here's a molecular biologist, Franklin M. Harold. He's an evolutionist. In his book, The Way of the Cell, 2001, he says we should reject, as a matter of principle, the substitution of intelligent design for the dialogue of chance and necessity. He's talking about these molecular machines. He says, we can't have this discussion about God. It's a matter of principle. We're scientists, is what he says. Of course, it's a ridiculous argument. He says, but we must concede there are presently no detailed Darwinian accounts, evolutionary accounts, of the evolution of any biochemical system, only a variety of wishful speculations. We know it's got to be, an answer's got to be out there somewhere. We know there's no God, and we know it all had to come about by chance. We don't have any idea how it is. All we have is wishful thinking. And that's kind of where we are on that, as far as an explanation for these kinds of things. We spent some time looking at the normal resolution level, at the amazing body that we have and all its systems. We looked at the eye and the ear and a number of other things. But do you realize something? None of this here in its normal resolution, everyday life we see it would work if it didn't have all these little machines behind the scenes. Not only did this have to evolve by some mysterious way, before that happened, all these guys had to evolve. Now, evolutionists don't like to do that. They talk about one or the other. In fact, the people who talk about the macroevolution that we see around us and the different creatures don't even go into the molecular biology very much. They just say, go talk to a molecular biologist about that. They ignore it. You know, I'm going to pick on Dan again. Dan drives up to my house and says, hey, come take a ride in this car. And so we drive around, and boy, it's cool. It just really fast, accelerates, brakes when we come to a stop, does what it turns the corners, comes back. And I go, this is a nice car. I like the way it looks. Let me check under the hood for a minute, just see what kind of engine it's got. And we pop the hood. There's nothing there. That's what evolutionists try to do with this, is that they talk about what's on the left, but they don't like talking about what's on the right here at all. I go, well, Dan, wait a minute. Where's the engine? I mean, I don't even know how we got around. There's no engine in here. There's no transmission. I saw you stop at the stop sign, but there's no brake cylinder here to run the brakes. There's not even any steering linkage under here. And I would go, Dan, and then I'd wake up, because that would be a dream, wouldn't it? If you had a car that drove around that didn't have the machinery necessary to run it. That's what evolutionists are trying to sell, unfortunately, is they talk about what's on the left, coming about by slow process, but boy, they get really afraid and scared. You'll see some of the quotes when we talk about these molecular machines and how they first had to come into existence before we could have everything we see now that we can see with our naked eye. Yeah, Spencer, wait for the microphone right there.
This point should not be overlooked. It's a powerful point, and it transcends into Bible study. One of the keys to error is what you just said, which is disconnection of information. Mm-hmm. It is a fundamental key to have error. If you look at one passage and ignore all the other passages, for example, baptism, very simple. If you look at all the passages on baptism, the vast majority of them don't actually mention water. But the ones that do connect them all together. Sure. But if you ignore the one passage or the two passages or the multiple passages just to focus on the one, you have the basis for error.
Yes.
And it happens in every area of our life. The key to error is disconnected information. So yes, we have the molecular biology. Oh yes, 13 billion years, that easily could have come about. But then don't talk to a mathematician. Just don't talk to a mathematician. And don't
Right.
talk to an astrologist, not an astrologist.
Astronomer.
Astronomer. Don't talk to that guy either, because when you talk to them together, they contradict each other dramatically.
Yeah.
And it all collapses.
Good point. Appreciate that. So life is even more complicated than so many evolutionists even want to admit. You've got a system of things, as we looked at when we were looking at the human body and different creatures, sophisticated mechanisms that have, like the bombardier beetle and his particular processes. Well, none of even that could have come about in whatever way you want to imagine until some of these things could come about before that support that whole physiology. Only answer there is, I will praise thee, for I am fearfully and wonderfully made. Marvelous are your works, and that my soul knows very well. I don't see how we could come up with any other
Amen.
explanation. Don't see how it's possible. Nothing else would work. You've got to be blind if you're trying to ignore all this evidence for why there's a God, for the evidence for why God does exist. Okay, so we're going to leave this part of molecular machines. We got a few minutes left. Let me see if we got time to start the next little thing here. Yeah, I think we do. We got 5 minutes. Information. Everything that works needs it. It's a product of intelligence. It's got to be obvious. Here's a fictional, let me just say this first. When someone sends a message, something rather fascinating and mysterious gets passed along. Think about it for a bit. A little fictional message that was sent off here. I'm going to use it. It's never really happened, but we're going to use this example as an illustration of what we're talking about. If you're sharp, you know what this is, some of this is about. I'm not sharp enough. I had to look it up. On April 9th, 1865, Richard, guy that we made up here, who lived in Boston, wanted to send an important message to his cousin, Ned, who lived out on the Nevada frontier. So Richard dictated the message to his secretary and asked him to send it by telegraph to Carson City, Nevada. From there, the message was taken to a nearby Indian village where one of the natives sent it out using smoke signals. Some 10 miles away, out in the wilderness, Ned looked up from his campsite and leaped for joy when he saw the message. Now, before we guess what that message is, which you might already know, let's make a point. Nothing physical has been transmitted. Not a single atom or molecule or any substance traveled from Boston to Nevada. And yet, something very real and very important traveled all the way. Does anybody know what that message might have been? Not even you historians. Whoa, I would have missed it. The war is over. That was the date the Civil War ended. And so here it was. Richard wanted to tell his brother Ned, who neither of those guys existed, but this is an example to explain a particular situation about information. Information is a marvelous thing. It doesn't have to travel in physical form from one place to another. The thing is, of course, we're not interested in that particular thing, but let's just notice this. Neither the sound waves from Richard's voice, they weren't the message. The paper and the ink that his secretary used to write that message down, that wasn't the message. That was just the paper and ink that transmitted it. The wire and electricity and the telegraph message, that wasn't the message. Or the smoke signals that went up that Ned could see, it wasn't the message. It's important to see this. These were only the materials on which the information was transmitted, and we see that. The only thing that could produce the information was a mind. That's the only thing that can produce information, is a mind. In this case, it was Richard's mind. And so we're going to see here next week as we get into this, genetic information which is stored in DNA is no different. Now, don't get afraid of DNA. DNA is just molecules like everything else, and I haven't shown the molecular form here because it wouldn't mean anything to me, really, and probably not much to you. But DNA is this coiled information storage system. It has 4 different letters: adenine, thymine, cytosine, and guanine. And they're arranged in base pairs, 2 at once, along this coil of DNA, and they mean something. There's information there that is used by the cell. If it wasn't there, you wouldn't have any life. DNA is not the information any more than the vibrating air caused by Richard's voice when he dictated his letter, or the paper and ink. That wasn't the message. DNA is not either. DNA only stores genetic information. The information had to come from somewhere else. Okay, next week, we'll come back, we'll look and see, could something like this have come here and come into existence by chance? Is there any way that could happen? So come back and we'll talk about that next week. Appreciate everybody's presence and your participation, the comments that have been made. Thank you for participating in that way. And we'll break for 10 minutes and get back at 10:30 for our worship service.